Hello everyone, this is Saman Aqeel, Assistant Professor IIMT College of Pharmacy. Today I will be discussing a very important topic that is controlled drug delivery systems. Moving on further, let's have a look on the definition of controlled drug delivery systems. A controlled drug delivery system or CDDS is defined as formulation that enables the introduction of a therapeutic substance in the body and improves its efficacy and safety by controlling the rate, time, and place of release of drugs in the body. Now, it is a formulation which is efficacious and safe, having a therapeutic substance that is released in the body, but the rate of the release of the drug is controlled. If the rate of the release of the drug is controlled, it is known as the rate controlled drug delivery system. If the time of the release of drug is controlled, it is known as the prolonged release or extended release drug delivery system. And if the place of release of drug in the body is controlled, it is known as the site specific or targeted drug delivery system. Now, the process includes three important steps. First step is administration of the therapeutic product, that is, administration of the prepared dosage form. Next is the release of active ingredient by the product, that is the release of the API from the dosage form. And the last step is the transport of the active ingredients to the site of action, the transportation of the API to the desired site of action. Now, these are some terms associated with the same. First is prolonged release or extended release dosage form. As I have discussed earlier, if the time of the release is modified or controlled, it is known as the prolonged release or extended release dosage form. There is no control on the rate or site of release, and the frequency of the dosing is also reduced because the time of duration of action has been enhanced, so the frequency of the dose is also reduced, and a constant level of the drug is maintained in the plasma. Next is the rate controlled drug delivery system. If the rate of the drug is controlled, it is known as the rate controlled drug delivery system. The rate of the drug is predetermined. The rate of the drug is predetermined. That is, the release of the drug is at a predetermined rate. It can be systematic, that is in the general circulation, or it can be local, that is to a specific site or specific period of time. And the last is the targeted drug delivery system or site specific. This is known as the targeted drug delivery system because the uh, drug is delivered to a specific tissue or a specific site, and the drug does not goes to any other non-targeted site. Now, this is the criteria for selection of drug. Some properties, physicochemical properties, physiological properties, or biological factors, these all are associated with the drug selection. And there are some physiological properties of the drug that need to be considered. That first is the aqueous solubility of the drug. It is very important that whether the drug is soluble in aqueous media or not. Second is the partition coefficient. It is very important. Partition coefficient means how much drug is soluble in organic phase and how much drug is soluble in inorganic phase. As we all know that all the membranes are made up of lipids, so it is very important to cross the lipoidal membrane for absorption or permeation. Third is the degree of ionization, that is the drug pKa, that is how much, what is the degree of ionization, what is the ability of the drug of being converted into its ion form. Next is the stability of the drug. Next is the molecular size and molecular weight of the drug. It is also very important parameters. The very large size molecules are not able to cross the small pores or the tight junctions of the membranes. And the last physiological property is the protein binding or the ability of the drug to bind with the protein. Protein binding is very important as it helps the drug to transport to different organs or different sites. Now coming on to the next, these are the biological factors which acts as the criteria for drug selection and also affects the efficacy of the drug. or the dosage form. First is the biological half-life, that is T half. What is the T half? T half is the how much time a drug is taking to eliminate from the body, 50% elimination of the drug from the body. Next is the dose size, how much milligram is needed in the body. Next is the therapeutic window, the safe concentration of the drug in the body, that is between the maximum effective concentration and maximum safe concentration. Next is the absorption window, and last is the patient physiology. It is very important that to know what is the physiology of the patient to select any drug for the CRDDS or controlled drug delivery system. Moving on to the next, we are having some synonyms which are given by various scientists for controlled drug delivery system. First is the smart delivery system. It was the term given by Shane and Robinson in 1982. Next is the targeted drug delivery system. It was the term given by Goldberg in 1983. Next is the intelligent drug delivery system. It was the term given by Weislu and Spicer in 1982. Next is the novel drug delivery system. This was given by Shane in 1982. And last is the therapeutic or rate controlled drug delivery system. It was given by Hellman in 1984. After discussing the important synonyms, let's come to the rationale. Rationale means the reason for the selection or for designing the controlled drug delivery system. In the earlier time or traditional era, we were having the conventional therapies. Conventional dosage form includes the simple tablets or capsules having immediate drug release and shorter duration of action. To cope up with this situation, we have designed the controlled drug delivery systems. Now, to show its therapeutic effects of a drug must achieve minimum effective concentration. Minimum effective concentration is the MEC, which is the minimum concentration of any drug to show the therapeutic effect or therapeutic action. Now, in the conventional therapy, the repeated dosing for the required duration, we have to give a repeated dosage forms for the required duration. Second is the fluctuating maximas, that is, the level of the drug was not maintained in the plasma. It was sometime higher, sometime lower. And next is the dosing interval is shorter than the biological half-life of the drug. That means 50% of the drug was not able to excrete from the body, and before that we were giving next dosage form. Shorter the biological half-life of the drug, higher is the frequency of the drug administration. That means if the half-life of any drug is shorter or less, we have to give multiple dose to maintain the duration of action. And last is the patient non-compliance and inconvenience. That means if we are giving a repeated dosage form to any patient, repeating doses has been given to patient, it must be non-compliant to the patient also. Now let's have a look on this diagram, which is showing the rationale of conventional versus controlled release. Here you can see on the x-axis time is given and on the y-axis plasma drug concentration is mentioned. You can see that the green diagram is showing the peak. There are two lines indicated with the pink border, pink line, dotted line. First is the MEC, that is the minimum effective concentration. And the upper one is the MSC, that is the maximum safe concentration. And between these, the area lies, it is known as the therapeutic level or the therapeutic index, therapeutic window. Minimum effective concentration is the minimum concentration of the drug which is being needed to show the therapeutic action, and the maximum safe concentration is the maximum concentration of the drug which is safe. After crossing to the MSC, that is maximum safe concentration, a drug may show toxic effect or toxicity. And if the drug is below the MEC, that is the minimum effective concentration, the drug will not show its therapeutic action. So we have to need some dosage form or some formulation that may act between the MSC and MEC, that is in the therapeutic level. Earlier in the conventional dosage form, the release of the drug was immediate, so we have to give multiple dose frequency. The dosing frequency was higher. We have to give a number of doses to maintain the duration of action. But you can see this graph, the green line is showing the sustained release. That means the release of the drug is being maintained for a longer duration of time, longer time in between the MSC and MEC. And this line showing the zero order control release. This means the release of the drug is zero order. That is, it is not dependent on the concentration of the drug. The release is zero order. The graph is straight, linear. The graph is linear, which is indicating that the release plasma drug concentration is constant, and it is maintaining in the body. After this, now come to the controlled drug delivery system. That means in a controlled drug delivery system, there are some important aspects. That is, first is the biopharmaceutical characteristics, second is the pharmacokinetics, and third is the pharmacodynamics. We have to make sure of all these three elements or aspects before designing the controlled drug delivery system. Pharmacokinetics means the effect of the drug on the body on the drug. Pharmacokinetics means the effect of the body on the drug, that is the ADME, absorption, distribution, metabolism, and excretion. And pharmacodynamics means the effect of the drug on the body, what the drug does to the body. Now, characteristics of a drug are optimized. These all three characteristics of the drug are optimized. Optimized means making it fully functional, or needing how much is essential, so as to attain a safe and effective therapeutic blood levels for a desired time period at the intended site. So our basic aim is to maintain a constant amount of the level of the drug in the plasma for a desired time period and to the intended site. Now, the system is designed to release the drug at a desired controlled rate as indicated by the controlled drug delivery system. The system is designed for the release of the drug at a desired controlled rate. The idea is to modulate the pharmacokinetics and pharmacodynamics of the API. The idea or the main concept behind the formulation of the CDDS is the modulation or the alteration of the pharmacokinetics and the pharmacodynamics properties of the API or the active pharmaceutical ingredients. Now, to make drug action more a dosage form design property and not the inherent drug properties. You can understand that a drug is having its own properties, and we are modifying or modulating the pharmacokinetics and the pharmacodynamic properties of the API. So after the modulation, we are coming towards the concept that the dosage form property, the property of the design property of the drug action must be shown by the modulated version, not by the original drug properties. The rate limiting step in drug release in the drug delivery and not in the physicochemical properties of the drug. The rate limiting step means the main step which is controlling the rate of the drug release. It should be in the drug delivery system, but not affecting the physicochemical properties of the drug. Now, this is a diagram which shows the drug release from conventional as well as from controlled drug delivery system. First, the drug is in the dosage form. After being released, it is in the solution form. And after the absorption and permeation, the drug is goes to the targeted site. The drug goes to the targeted site. Now, in controlled drug delivery system, the rate limiting step, that is, the rate limiting step is the release of the dosage form, release of the drug from the dosage form. And but in the conventional drug delivery system, the rate limiting step is considered to the absorption or the permeation, that after the drug is available in the solution form, how is the drug is absorbed, how the drug is being absorbed or permeated to the targeted site. Now, let's come to the advantage of these controlled drug delivery system. First advantage is that rate and extent of drug release and Hence, absorption can be modified to have a control of drug therapy. We are controlling the rate and extent of drug release, so the absorption is also modified, and the drug therapy having a controlled release. Next is the enhanced patient compliance and convenience, as is what discussed earlier. In the case of conventional drug delivery system, the patient was not compliance, the patient compatibility was not there, because there was a high frequency of dosage form. The patient has been given to a number of dosage form repeatedly, consequently, but here it is reduced, so the patient compliance has been enhanced. Third advantage is the efficient utilization of active agent. The API is being used efficiently. Improve safety margin of drug avoidance of fluctuating maximas and minimas in plasma level. This means that there is no fluctuation in the maximum safe concentration and minimum effective concentration in plasma levels. So the safety of the drug is improvised. Avoidance of sawtooth kinetic reduced fluctuations in side drug levels. Elimination of systematic side effects with high plasma concentration is also there. There is no side effect, elimination of the side effect of the systematic, that is the general circulation. Targeting of drugs eliminating damage to non-target tissues. This means we are giving the drug only to the target site, and there will be no damage to the non-targeted tissues. Now, if any system is having advantages, it may also have some disadvantages. These are: prompt termination of therapy is not possible in case of significant adverse effect. This means that if there is any significant adverse effect in the body, you cannot terminate the therapy immediately, because you are giving a large amount of dose in the body. Physician has less flexibility in dose adjustments. There will be less flexibility in dose adjustments. Designed on the basis of average biological half-life in normal populations, so variations like disease conditions are not accommodated, because it is designed on the basis of the biological half-life in normal population. The testing has been done in the normal population. Risk of dose dumping. Now, this is a very severe disadvantage of the CDDS. Dose dumping means the immediate or accidental release of the drug in the body, because you are giving a higher amount of drug in one dose, and if due to any accident the large amount of drug is released in the body, it may produce toxic effects or severe side effects. Reduced drug absorption may delay onset of action. This is very much understood. That is, if the drug absorption is reduced, there will be a delay in the onset of action of that drug. Variable drug absorption in individuals and at different times. This depends on the patient physiology, that there must be some variations in the drug absorptions among individuals and at different times. There will be poor in vitro in vivo correlation. In vitro means outside the body, and in vivo means inside the body. There will be a poor correlation between these in vitro and in vivo, because some things are different from practical things are different from theoretical concepts. All drug candidates are not suitable for making CRDDS. Not all the drugs are suitable for making these types of drug delivery systems. Now, there are a number of challenges also to these controlled release. First is the cost of formulation, because the preparation and processing is very essential process. We have to design these release at a predetermined rate. So the cost of the formulation is immediately enhanced. Fate of controlled release system if not biodegradable. This is a very important concept or aspect, that if the any of the polymer or any of the drug is not biodegradable, so we cannot say what will be the destiny or what will be the future outcome of this controlled release system. Next is the biocompatibility. We are not so sure about the biocompatibility between the drug, the body, and the polymer or the additives. Now, next is the same point is repeated here, but it is related to the polymer additives, that we don't know if the polymer are not biodegradable. we don't know what will the future of these polymer additives. And the last is the to maintain plasma concentration by releasing the drug at desired time. As I have told this earlier also, there is a very small difference, but there is a difference between the practical thing and theoretical thing. We are saying that we will be achieving this at a desired time, but it is little bit difficult to achieve this release at a desired time and to maintain the plasma concentration. But moving on further, by implementing some of the techniques, we are able to cope up with all these challenges to the controlled release system. Thank you for your valuable time.